Okay, Chapter 17, the focus of Chapter 17 is gene expression. In other words, from gene to protein. Now, there's a little more to it than that, but we're going to approach it from this angle here, from gene to protein. Now, make sure you watch the video and review your notes for Chapter 16. Information I covered on the structure of DNA and how we build the double-stranded DNA molecule is going to be relevant to what we cover here. So also make sure you go back to Chapter 5 and review the information we covered for nucleic acids and proteins, because you'll need that for Chapter 16 through 19. Okay, so in Chapter 16, we talked about DNA replication. In other words, taking original DNA, making identical copies of it. The original DNA is referred to as parent DNA. The newly synthesized DNA is called daughter DNA. And this occurs during S phase of interphase of cell division. What we're going to focus on in Chapter 17 is gene expression from RNA, or reading DNA, genetic information stored in DNA, reading that genetic information by a cell in order to make the RNA or protein that it needs. However, in this chapter, what I want you to focus on for gene expression is going from DNA to protein. We're going to exclude the RNA. I'll leave that for advanced classes. Now, this process called gene expression occurs whenever a cell needs RNA or protein. So again, in this case, we're going to focus on when protein is needed. So for DNA replication, if you reviewed your notes in the video, with DNA replication, okay, DNA is described as double-stranded. And that DNA that serves as a template to be replicated into new daughter strands, okay, we refer to that as parent DNA. That parent double-stranded DNA is given directional terms. Okay, if you're reading it from left to right, if the parent DNA strand is read left to right, 5' to 3', we call that the forward strand. And the forward strand is base pairs with the bases on the other single-stranded parent strand that we call the reverse strand. And in DNA replication, both parent strands, both the parent forward strand and the parent reverse strand, which are single strands each individually, are used as templates to replicate new daughter DNA. Okay. This is in contrast to what we're going to cover for gene expression. In gene expression, let me go back to this. In gene expression, both parent strands are not going to serve as templates, okay, for gene expression, only one of the two. But you have to know what I'm referring to when I say the reverse DNA strand versus the forward DNA strand. Now, gene expression, as I said, we're going to focus on gene expression whereby a cell reads, okay, genetic information stored in DNA. Okay, that genetic information comes in the form of units or regions of DNA that form genes. And these genes, I'm going to want you to think of in two ways. They code for polypeptides, which will eventually form active proteins, or another way to say it, they're simply protein recipes on how to make a polypeptide that eventually will become part of or form an active protein. Now, it's these proteins, if you recall, that do the work in your cells. It's also these proteins that are responsible for our phenotypes, the phenotypes of living organisms. What I'm showing you here is two different ways, okay, that proteins encoded by genes, remember genes are also known as alleles, two different ways these proteins, okay, impact your phenotype. In chapter 14, we went over Mendelian genetics and he was studying flower color. Eventually, the gene responsible for the color of a flower came to be known as a P allele because it existed in two forms. So you had an uppercase P allele, which coded for genetic information that led to purple flower color, and you had a lowercase P allele that coded for genetic information that led to a phenotype or a recessive phenotype, white color. In actuality, what it was, in the case of the dominant allele, the uppercase P allele, it actually codes for a protein that is an enzyme that produces a product called anthocyanin. Okay? This product called anthocyanin to our eyes appears purple. So it's molecules of this anthocyanin produced by a protein enzyme that leads to this dominant phenotype, purple flower color. So indirectly, okay, the protein is having an impact on the phenotype. However, if you recall, in lab, you worked with fluorescent proteins. And one of them you worked with was the GFP, green fluorescent protein. And it was found to be coded by a gene called the GFP gene. Okay. In this case, the protein coded for by this gene, the protein itself directly impacts the phenotype. Okay, so here you see a jellyfish fluorescing green. That's a phenotypic trait of this jellyfish, and this is directly being caused by green fluorescent protein. So in either case, either directly or indirectly, the protein coded for by the gene is having an impact on your phenotype. Gene expression itself, it doesn't matter if we're talking prokaryotic cells or eukaryotic cells. Gene expression itself is a two-step process. The first step is transcription. So notice in both types of cells, they show you transcription. The second step of gene expression is called translation. You can see for both of these, there's translation. Excuse me. However, if you notice over here for the eukaryotic cell, they're showing you they added something in you don't see in prokaryotes. It's called RNA processing. I'll explain that as we go through these steps. Okay. Keep in mind, besides a significant difference in size between prokaryotic cells and eukaryotic cells contain membranous organelles, such as the nucleus. All right, so we're going to factor that in. Therefore, when it comes to this two-step process, okay, this would be more closely resembling the two-step process. Again, we have a prokaryotic cell and a eukaryotic cell. But the way it works for this two-step process is in eukaryotes, since DNA is stored in a membranous organelle called the nucleus, okay, the first step called transcription, okay, has to occur and be completed, okay, before the second step, which occurs in the cytosol, can be performed, which is translation. So in eukaryotes, transcription and translation, these two steps, they're completely separate of each other. They happen at completely separate times. Transcription happens in the nucleus of a eukaryote. Translation happens in the cytosol. That's in contrast to prokaryotes. They do not have membranous organelles. Therefore, you will not find their DNA stored in a nucleus. Therefore, for prokaryotes, this two-step process of gene expression, transcription, and translation can actually overlap. However, it'll always begin with transcription first, and it'll always end with translation, but they're showing you here, okay, what they're trying to show you here is that transcription and translation do reach a point that they're overlapping. Now notice some of the terms in this drawing, okay. Prokaryotic DNA, first of all, it is double-stranded, but it's circular double-stranded DNA. Eukaryotic DNA, it is also double-stranded, okay, but it's linear DNA. Notice they're showing you the ends are open. So even though it's curved, it has open ends, all right. Notice next what they're showing you here, only in eukaryotes where you find the DNA stored in the nucleus, because prokaryotes don't have a membranous organelle. Notice this here, it's only pointing in this direction. We will talk about how in eukaryotes, when the first step is completed, transcription, we don't end up with a molecule of mRNA. We end up with a molecule called pre-mRNA, which stands for precursor, and I'll explain why as we get to that. Over here on this side, if you skip this here, notice it says mRNA and it's pointing in both directions. We'll talk about how for prokaryotes, this two-step process that forms gene expression, okay, it's more direct. It occurs more efficiently, whereas in eukaryotes, it's more complex. Then if you notice here, even though when we talk about transcription consisting of we talk about gene expression consisting of transcription and translation, and even though they say the final product is a protein, technically speaking, okay, after the second step, translation is completed, we actually have a polypeptide that is formed. So this is why I want you to go back to chapter 5 and cover proteins. When you cover proteins, make sure you cover the 4 levels of protein structure, because you're going to hear me mention that in the lecture today. And you can see here for translation, it occurs out here in the cytosol, which is the solution part of the cytoplasm, or in the case of the prokaryote, all we have is just cytoplasm or cytosol. This figure here is showing you a little more detail about transcription and translation, which I have on the first page. So the way the notes work is I give you a brief overview with a little bit of detail, and then we go into a lot of detail, beginning roughly on page 2. So here it's showing you that two-step process, transcription to translation. When transcription occurs, we go from DNA to RNA, not just any RNA, but we go to mRNA, which stands for messenger. There are going to be a total of 3 types of RNA involved in this process called gene expression, except we don't talk about the other 2 types till we get to translation. For translation, well, you got to think of it as translation in regards to different languages. Often you'll have a translator that translates one language to another language, such as Spanish to English, okay, or how do you say Japanese to English or Vietnamese to English, based friends that I have, and things of that nature. Well, translation has to do with, okay, going from one language in the form of nucleic acids or RNA nucleotides, translating that language to a different language having to do with proteins in the form of amino acids. So basically, we have a message in the form of RNA nucleotides being translated into a message of amino acids. And keep in mind, amino acids are your building blocks to form polypeptides, and then one or more polypeptides will form an active protein. Okay. Now, if you look up here, what else it's showing you? So this is a good figure I tell students if you want to print off or have on you, because it simplifies things, but it gives you a lot of detail too. Notice something up here. When transcription occurs, unlike DNA replication, they're telling you here, only one of the two strands serves as a template to make messenger RNA. That strand, if you notice, reading it left to right, is 3' to 5'. Therefore, this strand that's being shown, it is the reverse DNA strand, or the reverse parent strand. Oh, I was about to say I'm getting better, but I blew that right away. So it's the reverse parent strand that should be an R. Okay, just the reverse strand. Okay, the forward strand does not serve as a template. However, notice something when we go over this in more detail. This reverse DNA strand that serves as a template to synthesize mRNA. Notice the mRNA, they're giving direction. From left to right, it says 5' to 3'. That means it's in a forward direction, or it's a forward strand. For a geneticist, when they're studying genetic information at the level of DNA, reading the DNA to identify genetic information or identify genes, what they actually read is the forward strand. Because notice the forward strand, keeping in mind that for DNA nucleotides, one of the 4 bases is thymine. For RNA nucleotides, one of the 4 bases is uracil. Keeping in mind that U substitutes for T, notice this forward strand, even though it's not a template to make mRNA, it reads almost identical to the mRNA in the forward direction. Notice they're both 5' to 3'. So for scientists studying genetic information in the form of genes and DNA, they have to identify which of the two strands is the forward strand, because the forward strand, notice, reads almost identical to the mRNA. TGG, keeping in mind U substitutes for T, UGG. Down here, TTT, UUU, GGC, notice it says GGC, TCA, it says UGA. So notice they read almost the same. So again, when a geneticist is studying genetic information at the level of DNA, what they found primarily is the forward strand. However, it's the reverse strand that is used to synthesize a complementary strand of mRNA in the forward direction. Now notice with transcription is completed, notice that the mRNA, they're trying to tell you the genetic information carried or derived from DNA and being carried in mRNA, okay, it's coded. It's in the form of codons. So we'll talk about how once mRNA is synthesized, it is transported to a non-membranous organelle called a ribosome. And the ribosome, with some help, knows that when it reads the information, the encoded information on how to make the polypeptide, the ribosome knows it's not going to read one base at a time. It's going to read the mRNA, 3 bases at a time. These are referred to as base triplets called codons. So in other words, the ribosome, when it performs translation with some assistance, it'll read the mRNA 1 codon at a time. The reason for this is that each base triplet called a codon codes for one of the 20 amino acids, such as UGG. These 3 bases form a codon that codes for the amino acid tryptophan. UUU, these form a different codon that codes for a different amino acid called phenylalanine. If you look here, GGC, this base triplet forms a codon that codes for the amino acid, and this is one of the 7 you had to know in Chapter 5 called glycine. And then if you look here, they're showing you there's one more base triplet or one more codon, UCA, and this codon is code for a different amino acid, SIR or serine. There are many different codons that you will find, combined together forming a single molecule of mRNA. All right. So we'll talk about this codon and this codon table in a little bit more in depth in a bit when we get to that point. So again, like I said, make sure you go back to chapter 5, study nucleic acids and study proteins. Now, the way it works is once the let me go back one. Once the polypeptide is synthesized, at the end of translation, that one polypeptide may fold up into a tertiary structure that becomes an active protein. However, for many active proteins, they're made up of 2 or more tertiary structures that have combined together, forming a quaternary structure. Okay, so once that polypeptide is synthesized, it may or may fold up and become an active protein. It may fold up and become one of several tertiary structures that forms a quaternary level protein that then becomes active. Case in point, the molecule hemoglobin. The molecule hemoglobin is made up of 4 tertiary structures. 2 of them are referred to as alpha chain, 2 of them are referred to as beta chain. If you look closely, they're showing you each of these looks like a little twisted wire that's twisted up. Well, each of these little twisted wires that's a different color represents an individual polypeptide that is folded up into a tertiary structure. And all 4 of these tertiary structures have complexed together or come together, forming a quaternary structure. Most of these molecules called hemoglobin that are produced and found in red blood cells, most of the molecule is protein, but not all of it is because it has some iron in a group called a hemigroup. But you can see here, it's made up of 4 tertiary structures that have complexed together. Okay. Each of these tertiary structures is made up of a single polypeptide. Now, often instead of saying tertiary structures, we would say protein subunits. So often what you'll hear is hemoglobin is made up of 4 protein subunits. 2 of them are alpha subunits, 2 of them are beta subunits. And this is showing you here that the genetic information that codes for these polypeptides that form hemoglobin, they're not located. Their gene loci is on different chromosomes. Okay. So going into gene expression, starting with the first step, transcription. Okay. This is general information, and then we'll go into details. Listen carefully. It's very important. The enzyme, okay, the protein enzyme responsible for catalyzing this process called transcription is RNA polymerase. Sometimes it's called RNA pol, just simply POL instead of writing out polymerase. Do not confuse this enzyme with the 2 enzymes that were talked about in chapter 16 for DNA replication, which are DNA polymerase. Okay, listen carefully though. Whenever an enzyme has as part of its name polymerase, automatically tell yourself not only is it an enzyme because it ends in ASE, but it's an enzyme that requires a DNA template. So keep that in mind. Whenever an enzyme is given as part of its name, polymerase, it's not only an enzyme, ASE, but the polymerase, the overall term, refers to an enzyme that requires a DNA template to synthesize, a polynucleotide. The first part of its name lets you know what it's going to synthesize, what type of polynucleotide. Therefore, if it's a DNA polymerase, it requires a DNA template to synthesize a DNA polynucleotide. But for gene expression, transcription, the enzyme is called RNA polymerase. It also requires a DNA template, but the first part of its name tells you what it's going to synthesize, an RNA polynucleotide. Okay? RNA polymerase, unlike our DNA polymerase, in particular 3, doesn't require a primer. It does a whole lot on its own. Also, as it's showing you from here to here, RNA polymerase only uses one of the 2 strands as a template. It only reads one of the 2 strands or uses it as a template to make mRNA. If you look up here again, 5' to 3' left to right, that's the forward strand. 3' to 5' reading it left to right, that lets you know it's a reverse strand. Notice down here, they're showing you RNA polymerase would use the reverse strand to synthesize a molecule of mRNA in the forward direction. And that's what it's telling you down here. Now listen carefully, something I didn't stress enough at the beginning. DNA in the cells of living organisms has always been found to be a double-stranded helix, whereas RNA in living organisms has been found to exist as single-stranded. And this is showing you right here. Okay. Now, will that single strand of RNA base pair with DNA or fold up on itself in base pair? Yeah, you can see that, but it's always usually and only temporary. So notice this here. We'll talk about that in a little bit. How this mRNA that's being made using the reverse DNA strand as a template, notice it's not staying attached. It's coming off. Okay. RNA polymerase is the enzyme that catalyzes this first step called transcription. Okay. But transcription itself is broken up into 3 basic substeps. Okay. But these substeps all involve RNA polymerase. So what these 3 substeps are initiation, elongation, termination. Now, before I go over details here, I want to talk about this because this is the simplest figure to first go over this. Okay. RNA polymerase, it doesn't matter if it's prokaryotic RNA polymerase or eukaryotic RNA polymerase. RNA polymerase was found not to be able to locate the gene directly. RNA polymerase can't locate the gene directly. It needs help. It was discovered that genes found in both prokaryotes and eukaryotes are bracketed by 2 types of DNA regulatory sequences. These DNA regulatory sequences are simply known as promoters and terminators. Now they say promoter DNA and terminator DNA, but they're known as promoters and terminators. And as I said, these are DNA regulatory sequences that bracket the gene. In other words, the promoter is found upstream of the gene or right before the gene. And then there will be another region of DNA that forms a DNA regulatory sequence located just after the gene. That will be the terminator sequence. Okay? So I mentioned this in lab 9, day 1, that whenever they want to create, scientists wanted to create a GMO, they discovered it's not enough to identify a gene that codes for a particular protein that will enhance the traits of the organism they want to modify. They found that even when they isolate the gene that codes for that protein, that gene is useless when they put it into the new species cells, because they have to make sure that gene is bracketed by a promoter and a terminator. The reason for that, RNA polymerase is the one responsible for catalyzing this first step of gene expression. If transcription doesn't happen, translation doesn't happen. And RNA polymerase cannot initiate transcription if there is not a promoter, okay, a DNA regulatory sequence called a promoter, to help RNA polymerase not only locate the gene, but locate the start point of the gene. So they're showing you here, RNA polymerase actually recognizes and binds to the promoter. And when it recognizes and binds to the promoter, that not only helps RNA polymerase locate the gene, but if you go down here, it helps RNA polymerase locate the start of the gene so that it can initiate transcription. Elongation simply means synthesizing the molecule of mRNA by RNA polymerase. So if you look here, once initiation has been completed and our RNA polymerase has located the promoter and slid over to the start of the gene, what RNA polymerase will do is unwind and separate the double-stranded parent DNA, creating a transcription bubble. Then RNA polymerase will locate which of the 2 parent strands is the reverse DNA strand and uses it as a template to synthesize mRNA. Then what RNA polymerase will do is it will continue to move what we call downstream or in the direction of the terminator, and it will continue to unwind and separate the parent double-stranded DNA so that it can continue to use the reverse strand to synthesize the mRNA. The catch is this. This transcription bubble never elongates. It never grows larger. You will only find the transcription bubble wherever RNA polymerase is located. So notice it's only where RNA polymerase is. The reason for that is because RNA polymerase does not want to permanently separate the double-stranded parent DNA. As it performs elongation, the DNA that it's already transcribed, making mRNA, it will release it, and the two single strands here will passively reanneal. In other words, they will passively base pair, forming hydrogen bonds, and we passively get our double-stranded DNA reformed as it goes down. Eventually what will happen is the mRNA will reach a DNA regulatory sequence called a terminator sequence. This terminator sequence, part of it or all of it, will actually be transcribed and become part of the mRNA molecule. However, as RNA polymerase is transcribing this terminator sequence, it's reading the terminator sequence. And when it reads the terminator sequence, that lets RNA polymerase know, now I need to proceed into the third basic step, termination, which simply means stop synthesizing the mRNA. Release the parent double-stranded DNA and release the mRNA so that the cell can transport it to where it needs to from there. So these are the three steps. Now the details of this. There's a slight difference between the first step initiation in prokaryotes versus the first step initiation for transcription in eukaryotes. It's a little more complex in eukaryotes. For prokaryotes, there's only one type of RNA polymerase, and this RNA polymerase does a whole heck of a lot on its own. On its own, prokaryotic RNA polymerase is able to locate and bind to the promoter. On its own, it's able to slide over, and they're not showing it here, but it's able to slide over to the beginning of the gene and unwind to separate the double-stranded parent DNA, creating a transcription bubble. That unwinding and separating of the double-stranded parent DNA, creating a transcription bubble, they simply refer to that as unzipped. The DNA is unzipped. I don't like that term so much because, well, I basically never saw it or heard it from my professors or any research articles that we went over. Up here where it says transcription unit, what they're referring to up here is simply the gene, everybody. So just to let you know, if you see transcription unit, just tell yourself that means gene. Right. So again, this first substep of transcription, this basic step in initiation, prokaryotic RNA polymerase carries it out itself. It locates the promoter, it slides over to the beginning of the gene, the start of the gene. Okay, so it can initiate transcription. In eukaryotes, however, first of all, there's not one type of RNA polymerase. There's three types. RNA polymerase 1, 2, and 3. Okay, two of these three RNA polymerases are responsible for synthesizing molecules of RNA in general, and I'm not going to go over that. The one we're interested in is RNA polymerase 2. It's RNA polymerase 2, eukaryotic RNA polymerase 2, that synthesizes messenger RNA. This is the only one of the three RNA polymerases that synthesizes mRNA. The other types of RNA polymerase synthesize other types of RNA that are not called mRNA. Okay, so that's one difference with prokaryotes. There's not one type of RNA polymerase as there is in prokaryotes. There are three types in eukaryotes. Another difference, okay, is that this RNA polymerase 2, it can't locate the promoter on its own. It needs the help of proteins called transcription factors. So these transcription factors are shown right here, as they're showing you here. And they even put it here. So transcription factors, these are proteins. Okay, and since they play a role in this first step of gene expression called transcription, they simply call them transcription factors. But technically speaking, there are many different types of transcription factors, but they all influence RNA polymerase. In this case, they influence RNA polymerase 2 by helping RNA polymerase 2 locate the promoter. Eukaryotic RNA polymerase 2 cannot locate the promoter on its own. It needs the help of transcription factors. These transcription factors are responsible for locating a promoter next to a gene, and then recruiting, or in a manner of speaking, calling over RNA polymerase II, saying, hey, look, here's a promoter so that we can help you locate the gene you need to transcribe whenever the cell tells you to. So here you can see, with the help of the transcription factors, RNA polymerase II has located the promoter, slid over to the start of the gene, created a transcription bubble so that now it can initiate transcription. Okay. Listen carefully. On the test, since I'm not going to test you on RNA polymerase 1 and 3, for the test, you can know eukaryotic RNA polymerase 2 as just simply eukaryotic RNA polymerase. So again, one more time. For the test, for the final exam, I'm not testing you on RNA polymerase 1 and 3. Therefore, you can know eukaryotic RNA polymerase 2 as just simply RNA polymerase. Okay, so once initiation has been completed, that first basic step, the second basic step can occur now. Elongation. First of all, elongation is continuous. Again, if you watch the recording on Chapter 16, DNA replication, okay, the synthesis of the new daughter strand is not always continuous. All right, it could be discontinuous or continuous, leading to a lagging strand or a leading strand of daughter DNA. That's not the case for the transcription substep 2 elongation. Okay, when RNA polymerase synthesizes mRNA, it is a continuous process. There are no fragments being made. It's just one shot across. Notice here how it's showing you. Okay, the transcription bubble is only located wherever RNA polymerase is attached and unwound the DNA. Once it finishes transcribing a stretch of the reverse strand, as it moves down, it'll keep unwinding and separating DNA, so that transcription bubble is moving along with RNA polymerase. However, the DNA or the reverse strand that's already been used as a template to synthesize mRNA, once RNA polymerase lets it go, the forward and the reverse parent strands will automatically reanneal. In other words, they will automatically rebase pair, forming hydrogen bonds, reforming that double-stranded structure. So if you notice here, RNA polymerase reads only one strand. The mRNA that it synthesizes complements the reverse DNA strand. So what they've done is they've taken this here and they've magnified it. Here's our forward strand in light blue. Notice it's not being used as a template. Here's our reverse strand. Notice RNA polymerase here, which is depicted as a large blob shaped like a guitar pick. Notice it's only using the reverse strand. Okay. Again, what it's simply doing is RNA polymerase is reading the bases. Listen carefully. If you notice the figures I've shown you so far for DNA and RNA, okay, when they don't show you this, when instead they show you a figure like this with more detail, notice they're only showing you the bases. But these bases are part of DNA nucleotides or RNA nucleotides. However, as you review that information, if you haven't in Chapter 5, okay, for DNA nucleotides, there's 4 types. All 4 types consist of a sugar, deoxyribose, a negatively charged phosphate group, and any one of 4 bases, A, G, C, T. For RNA nucleotides, they also consist of a negatively charged phosphate group, the sugar ribose, and any one of 4 bases, A, G, C, U. But the key to genetic information is in the bases. Okay, the little strap here that they're showing you in this figure, that represents the sugar phosphate backbone. Okay, covalently bonding one nucleotide to the other for both DNA and RNA. Okay, but the reason why they're definitely making the bases stand out has to do with the key to genetic information is in the bases. This is why there was so much controversy, okay, about scientists accepting the fact that genetic information was stored in DNA. DNA was too simple. When you look at the complex structure of DNA, that double-stranded helix, the only real difference are the 4 bases. There's no way that 4 different bases could lead to all the complexity of the different living organisms or different species. But basically it fell under less is more. Even though there's greater amount of diversity in proteins, it was discovered that DNA, okay, less is more. Those 4 bases actually did lead to a high degree of complexity. Therefore, genetic information is stored in DNA, and the key to it is the bases. So notice when RNA polymerase is synthesizing mRNA, it's reading the bases specifically in the reverse parent strand. And wherever it sees the base thymine in one of the DNA nucleotides that forms the parent reverse strand, RNA polymerase will grab an RNA nucleotide with the base cytosine because with the base adenine because adenine complements thymine. When it moves down to the next DNA nucleotide and it sees the base adenine, it'll grab an RNA nucleotide with a complementary base, but in this case, it's uracil because we have U instead of T. Notice how these bases complement each other. So it's just reading the bases in the DNA in the parent reverse strand, and then that will determine what type of RNA nucleotide it will grab. So in other words, it's synthesizing the complement, but in the form of RNA. Again, as it moves past or as it finishes transcribing part of the reverse parent strand, what it'll do is it'll continue to slide down, unwinding and separating parent DNA, creating a transcription bubble, and that reverse template strand that it's already used to synthesize mRNA, it will not only let it go, but in a manner of speaking, it will kick off the mRNA it's already synthesized so that the reverse and forward parent strands can reanneal, reforming that double-stranded helix as it continues to slide down, performing this process called elongation. Eventually, downstream of the gene or on the opposite side of the promoter, okay, on the opposite side of the gene, opposite of the promoter, what eventually happens, okay, is that RNA polymerase will reach the terminator sequence. In both prokaryotes and eukaryotes, the terminator sequence is going to be transcribed, but there's a little bit of a difference. In prokaryotes, they simply say the terminator sequence is transcribed, which it's not showing you here, but it is transcribed. And when it's transcribed, as it's being transcribed, that's when RNA polymerase is reading the bases in the terminator sequence, and it's realizing, okay, I finished synthesizing the molecule of mRNA. I can release the parent DNA, and I can release that molecule of mRNA. In eukaryotes, the terminator sequence is often referred to as the polyadenylation signal, but it is transcribed, okay? And again, it also lets RNA polymerase know as it's transcribing this terminator sequence called the poly signal, it's letting RNA polymerase know, hey, you're through. Terminate this process called transcription. Stop the process. Release the parent DNA, release the mRNA, you're finished. Except in eukaryotes, I didn't put it here and I should have. Except in eukaryotes, the RNA that is made is not called mRNA. It's referred to as pre-mRNA. And I'll show you this in a second. I've got it a little bit better written later. So it's pre-mRNA. So these are the 3 steps of transcription. Now the way it works is in prokaryotes, once that one and only type of prokaryotic RNA polymerase completes these 3 substeps that form transcription, okay, what it's synthesized is a molecule of mRNA that is ready to be translated. Well, if we go back, okay, I didn't realize I went back that far. If we go back, keep in mind, okay, technically speaking, as that one and only type of RNA polymerase in prokaryotes is performing the 3 steps of transcription, okay, technically speaking, as it's continuing to perform the 2nd step, elongation, once enough of the mRNA has been made, it's ready for translation. So it's a much more straightforward process in prokaryotes, and it occurs much quickly since at this point it's during the 2nd substep of transcription, elongation, that once enough of the mRNA has been made, that the 2nd step translation can begin and you have the overlap. In eukaryotes, it's not true for 2 reasons. One, DNA is stored in the nucleus. Okay, so when the mRNA is being made, even if that mRNA was ready translated immediately, the membrane of the nucleus, nuclear envelope, is in the way. So what occurs though is what is found out. That's one reason why the two steps cannot overlap in eukaryotes. There's a second reason why the two steps can't overlap. In eukaryotes, when RNA polymerase finishes the three steps of transcription, what is synthesized is not the final product mRNA that's ready to be translated. What scientists discovered is once RNA polymerase, eukaryotic RNA polymerase, has completed the three steps of transcription, what has been produced is pre-mRNA. It needs to be modified before it's ready for the ribosome to translate it. If the cell were to transport this product produced by RNA polymerase, pre-mRNA, if it were to just transport it immediately to the ribosomes, they wouldn't be able to do anything with it. Okay, so what we're going to do now is in your notes for eukaryotes. Okay, I edited the notes, so if you looked at them early, I just re-edited them this morning. What I put down is at the end of transcription, okay, pre-mRNA has been synthesized in eukaryotes, and it must undergo 3 post-transcriptional modifications, which they're showing you some of them here, before it is considered the final product mRNA. So notice in this figure, pre-mRNA, we're going to talk about the 3 modifications it goes through before we actually consider it mRNA, comparable to prokaryotic mRNA that is ready to be translated. Okay, so this is a slide here, and I also edited this part of the page. I put down first, second, and third to make it stand out. Okay, what will occur is in eukaryotes, when RNA polymerase completes transcription in eukaryotes, what has been synthesized is pre-mRNA, and that pre-mRNA needs to be modified, okay, into the final product or finished product called mRNA that's ready for translation. This pre-mRNA undergoes 3 types of modifications. The first type of modification is to the 5' end of the molecule of mRNA here, pre-mRNA here, will be a 5' cap that is added. So what I want you to know this as is just simply the 5' cap. If you take a genetics class or an advanced cell and molecular cap, they will actually call it the 5' methylguanine cap. But I just want you to know it as a 5' cap. It's made up of a special type of guanine nucleotide. So that's what they're showing you here. Okay. This 5' cap is required to help attach the molecule of mRNA to the ribosome. So that's the purpose of this 5' cap. However, a second modification is needed. If you recall, I said the terminator sequence will be transcribed, but the terminator sequence is better known as the polyadenylation signal. There is a protein, an enzyme, okay, that after the pre-mRNA has been synthesized, this protein enzyme will come up to this pre-mRNA and it'll recognize this combination of bases. I will not test you on this, okay? What they're showing you here is that these 6 bases form the signal called the polyadenylation signal. There is an enzyme that will come up to the mRNA, scan the base sequence in the mRNA, and when it comes across these 6 bases, that enzyme recognizes that as a polyadenylation signal. And what the enzyme will do is it will add a poly-A tail. So going into the test, know this as a poly-A tail. But notice the poly-A tail is not added to the 5' end, it's added to the 3' end. Okay? So the first modification is the addition of a molecule that I want you to know is a 5' cap. It's added to the 5' end of the mRNA. The second modification, it's the addition of a poly-A tail. It is added to the 3' end, adjacent to the terminator sequence known as a polyadenylation signal in eukaryotes. Now, the poly-A tail, the reason why they call this the poly-A tail is if you look here. What happens is the protein enzyme, when it recognizes this signal, okay, it will add, okay, a strand of, it will add a polynucleotide strand made up of only the bases adenine. So in other words, you'll have many RNA nucleotides covalently bonded one to the other. All of those RNA nucleotides will have the base adenine. Thus, we call it a poly A tail. This poly A tail could be formed by 50 RNA nucleotides, all with the base adenine, or it could be formed by 500 RNA nucleotides, all with the base adenine. Therefore, they simplify it and simply call it the poly A tail. I haven't looked at the recent literature, but at one time, I know in the 90s, the belief was the longer the poly A tail, the longer the mRNA will stay present so that the ribosome can translate it over and over and over again. The shorter the poly A tail, the quicker the cell will remove it. In other words, that's one way a cell will control how many polypeptides are for the mRNA. The last modification, okay, is simply called RNA splicing. For eukaryotes only, and I'm going to go to the next slide here. For eukaryotes, what was discovered is that in units of DNA that form genes, so again, it says RNA coding sequence, but what they're referring to is a gene. So as complicated and as terrible as you might think I'm doing here, trust me when I tell you, this is basics. It gets more complicated. So even if something's not making sense, if you can at least remember it and remember where you saw it in a figure, hopefully when you see it again, it'll start making more sense, okay? So if you look, what they found is for those regions or units of DNA that form a gene, okay, in eukaryotes, that eukaryotic gene isn't made entirely of a coding sequence that codes for a polypeptide. What they found is that there are small stretches within the overall region of DNA that forms a gene that consist of non-coding segments that do not code for the polypeptide. Okay, so what happens is in eukaryotes, when RNA polymerase performs transcription, making pre-mRNA, those stretches of mRNA that contain the actual coding sequence for the polypeptide, those stretches are referred to as exons. So simply put, exons, okay, contain the coding sequence for the polypeptide. In other words, that's what the ribosome will read so it knows how to make the polypeptide. So this is what I've circled here. So when pre-mRNA is made, in between 5' cap and the poly A tail, there are only stretches of messenger RNA or pre-mRNA that actually contain the coding sequence for how to make the polypeptide. We call these exons. In between these exons are stretches, okay, are stretches of sequence that are non-coding. We call these introns. So introns contain non-coding sequence. In other words, the intron does not code for the polypeptide. So again, in other words, the intron does not code for the polypeptide. Therefore, the third modification that has to occur is RNA splicing. Simply put, RNA splicing has to do with removing these introns. So the introns are removed during RNA splicing. And what happens then is that the same enzyme that removes these introns during RNA splicing will reattach the exons to each other. So now what we have are all three of these exons, after splicing occurs, all three of these exons will be reattached into one continuous polypeptide coding sequence. It says protein, but polypeptide coding sequence. Okay, so we call this RNA splicing. RNA splicing occurs in eukaryotes. It's a third modification. This has to do with where introns are cut out and the exons are reattached. So now we finally have our molecule we finally have our final product, mRNA. And all it contains is continuous coding sequence for the polypeptide. So now the ribosome can read the genetic message being transported by mRNA to make the polypeptide. There is an enzyme responsible for this third modification called RNA splicing. It's actually an enzyme complex. It's called a spliceosome. So a spliceosome is simply known as an enzyme complex. So the abbreviation for enzyme is ENZ, and a spliceosome is an enzyme complex. What they're showing you on this figure, and I probably need to rearrange the PowerPoints a little better. When it's in person, it makes sense the way I have the PowerPoints arranged, but online it's not. I think I'll rearrange it some. But if you look here, it's showing you up top. We have our pre-mRNA, an exon 1, intron, and exon 2. So this intron is separating these two exons. This enzyme complex called a spliceosome, it performs RNA splicing. It recognizes the non-coding sequence in mRNA, in the pre-mRNA, called an intron, and it will cut it out and remove it. So here it's showing you the intron's been cut out by the spliceosome. And then what the spliceosome will do is it'll take these exons, in this case exon 1 and exon 2, and it'll covalently bond them together. And now we have our final product, our mRNA, and you can see here the coding sequence on how to make the polypeptide. It's uninterrupted. It's now continuous. So then the question is, why? Well, the way it worked, okay, initially, when scientists discovered this, is they said, why is it so much more complicated in eukaryotes than prokaryotes? Because in prokaryotes, when our polymerase finishes transcription, the mRNA is made. It's ready to be translated. It reads continuously. The genetic code reads continuously on how to make the polypeptides, such as redheads are beautiful. But in eukaryotes, when RNA polymerase completes transcription, we have pre-mRNA. The genetic code, the code on how to make the polypeptide, it's broken apart by introns depicted by blah, blah. So they were wondering, why would this happen? At the same time, okay, at the same time, from the 80s into the 90s, into the late 90s, okay, scientists were discovering that the number of proteins produced by the cell or cells of a living organism did not equal the number of genes stored in the genetic information of DNA. So when I was an undergrad, I had a professor say there are at least 100,000 genes in the human genome, because there were at least 100,000 different proteins that had been identified that do various types of work. When I was in grad school, sitting in a class, the professor says there are roughly 50,000 genes in the human genome. And there are way more proteins that have been discovered that do different types of specific work. So it wasn't matching up. Then the sequencing of the human genome occurred. And when that was completed, they said, oh my God, we don't have 50,000 genes. We maybe have 20,000 genes in the human genome. And by this time, way more than 100,000 different proteins that do specific types of work had been discovered. It wasn't matching up. So scientists were confused. They were baffled. How is this possible? Well, what they discovered is not only does RNA splicing occur, but sometimes the genetic information stored at the level of DNA, okay, that genetic information, when it's transcribed into pre-mRNA, that pre-mRNA is sometimes very simple pre-mRNA, where when RNA splicing occurs, the areas here that say blah represent the introns are spliced out, and then we end up with redheads are beautiful. In other words, the exons are put together. But what they found out is that sometimes it's not that simple. Somehow, some way, the cell knows, okay, that sometimes the genetic information stored at the level of DNA, when it is transcribed into pre-mRNA, the cell can actually alternatively splice that pre-mRNA. In other words, when they would look at the pre-mRNA, they would sometimes notice that, yeah, we still have our exons and introns. Again, the introns depicted by blah, blah. But when they looked at the exons, they started to realize, wait a minute, The exons contain coding sequences for how to make the polypeptide, but not all of these exons are necessarily needed, okay, when pre-mRNA is converted to mRNA. They call this alternative splicing. In other words, sometimes when genetic information is transcribed into a pre-mRNA, okay, and you look at it, the exons read in a manner that you can recombine them during the process of splicing. In other words, this pre-mRNA could be spliced to where it reads redheads are totally beautiful, or it could be spliced to where it says brunettes are not beautiful. Please, I'm just saying, I don't have any hair, so please take it easy here. Or it could be spliced to where it says blondes are totally not beautiful, or blondes are totally beautiful, or the same thing for brunettes and redheads. Therefore, it could be alternatively spliced. What I'm getting at here is that because not only does RNA splicing occur, but because sometimes some pre-mRNAs can be alternatively spliced, this means a single pre-mRNA could be spliced into 2 or more different types of mRNA that are translated into 2 or more different types of polypeptides. So in other words, one gene, which is transcribed into one pre-mRNA, could actually lead to the production of 2 or more different types of active proteins. So it gets very complicated is what they're finding out. So basically what happened is at the completion of the Human Genome Project, it got very quiet. And I had finished grad school by then, but those of us that were in the area of genetics and stuff, we were laughing because we were saying, yeah, it ain't that simple after all. They thought they were going to be able to do all these wonderful things when they sequenced the human genome, especially the genetic information stored in the DNA, and they found out, no, it's actually much more complicated than what they thought. Okay, what I'm going to transition into now is translation, okay? And I should have put that up here at the top. Now, translation has what I call more working parts. So the way I approach translation is first by reviewing the molecules and organelles that are needed to perform translation. Okay, so in your notes I put down what is required to perform translation. First are 3 different types of RNA. We've talked about one, mRNA. The M simply stands for messenger. What message is it carrying? Well, the genetic information on how to make a polypeptide. Okay, but there are 2 other types of RNA that are needed for translation to be completed. These 2 other types, sorry about that. These 2 other types are transfer RNA, which we call tRNA, and the third type, which we only briefly mentioned, and I forgot to put it on here, is ribosomal RNA. So the R stands for ribosomal RNA, or simply rRNA. Okay, these are the 3 types of RNA that we need. The other component that we need, okay, to perform translation, and it was shown there, is simply a ribosome. If you recall, we talked about organelles in chapter 6. You can go back and look at that. And we said the function of a ribosome is to synthesize proteins, but we're going to be a little bit more specific. Okay. In this chapter, we're going to talk about how ribosomes synthesize polypeptides through the translation of mRNA. So this is what we're going to focus on. Okay. All right. So translation, the second step. Translation occurs at the ribosome. This is where translation occurs. It occurs at the ribosome. This is why the ribosome is given credit for protein synthesis. It occurs at the ribosome, but technically speaking, the ribosome can't do it on its own. It needs help. Okay, the messenger RNA, mRNA, it's carrying the message on how to make the polypeptide in the form of codons. Okay, but it's going to need assistance. First of all, just structurally speaking, we'll talk about how a ribosome needs RNA. But also when it comes to translating the message in mRNA, it's going to need the help of another type of RNA you're going to come to know as tRNA. So again, we talked about this a little bit. mRNA, the M stands for messenger. What message is it carrying? Well, It's carrying a genetic message. It's carrying a message from a gene derived from DNA on how to make a polypeptide. It's transporting it to the ribosome. It's a lot easier than basically grabbing this huge, either circular piece of double-stranded DNA in prokaryotes, or trying to pull out of the nucleus, okay, one of 46 chromosomes, or a pair of 46 chromosomes out of the nucleus of a eukaryotic cell. It's much simpler to just make a copy. You may have done this. Okay, when you're in college, you have a lot of large textbooks. Sometimes you don't want to lug the textbook around. You might just write down some of the information and take it with you. Now, as I said before, the genetic information derived from DNA, okay, that is being transported in mRNA to the ribosome, okay, that genetic information is in the form of codons. And these codons are referred to as base triplets, okay. Each codon codes for either a specific amino acid or it's called a stop codon. So here it's showing you mRNA contains a genetic code in the form of codons, and it's showing you all the different codons that exist. Now keep in mind, how many different amino acids are there? There are 20 different amino acids. And what was discovered is that these codons can be redundant. Okay, so here it's talking about the codon, and these codons can be redundant. What they mean by redundant, well, I've given you a couple. Okay, the only codons, first of all, the only codons you're responsible for on the test are the ones in your notes. Some of the ones I've given you, okay, have to do with tyrosine and serine. These are two examples of showing you what we mean by redundant. If you notice here, the ones I've given you are not even all of them for serine. But if you look at tyrosine, what we mean by redundant is that there are two different codons that code for tyrosine that you can find in a single molecule of mRNA. The ribosome doesn't care if the codon is UAU or UAC in that molecule of mRNA. When the ribosome sees either one of these codons, it knows it needs to grab an amino acid tyrosine. Likewise, if you look at serine, I only gave you two, and those are the only ones responsible. But notice something. To really emphasize what we mean by redundant, notice for serine there are up to 6 codons. Okay, I want you to know just 2 of them. But the bottom line is, if any one of these 6 codons is in a molecule of mRNA, or any one of a combination of these 6 codons is in a single molecule of mRNA, that lets the ribosome know it needs to grab the amino acid serine. Okay. Now, is this true for all codons being redundant? No. There is only one codon that they call a start codon. That codon is down here. Okay. There is a codon simply called AUG, adenine, uracil, and guanine. So you have 3 RNA nucleotides. One has a base adenine, one has a base uracil, one has a base guanine. These 3 RNA nucleotides form the codon AUG, and this codon is known as a start codon. What I mean by start codon is this codon tells the ribosome where to begin translation. So again, this codon AUG is called the start codon because it tells the ribosome where to begin translation. Well, this codon, okay, codes for the amino acid MET. This is also one of the 7 you were responsible for knowing in Chapter 5. MET is short for methionine. Therefore, in eukaryotes, this is just for eukaryotes. In eukaryotes, I'm sorry, I forgot to mention that. In eukaryotes, whenever translation begins at the ribosome, the first amino acid grabbed by the ribosome is methionine. Okay. Now, whether or not that methionine becomes part of the final product, the active protein, that's another thing that's more involved. So you can see here, the start codon is called AUG. It codes for the amino acid methionine. And notice, this amino acid methionine, there's no redundancy. This is the only codon that codes for it. But that's still not enough. It could be that in a single molecule of mRNA, there is more than one start codon. more than one AUG codon. So how would the ribosome know which one is the start one? In your notes, I gave it to you. Okay. The start codon, AUG, is embedded in a larger mRNA sequence called the Cossack sequence. So in your notes, I put down Cossack sequence. If you look at that Cossack sequence, which is made up of 7 bases, underline the 3 bases that read AUG. So that's how a eukaryotic ribosome is able to locate the start codon. It is looking for the AUG codon embedded in a larger RNA sequence called the Cossack sequence, and that lets the ribosome know that is the start codon. That is where I will start the process of translation. So in other words, just like RNA polymerase needs to know where the beginning of a gene is to start transcription and needs to know where the end of the gene is to terminate transcription, a ribosome also needs to know, where do I start translating, okay, the mRNA, and where do I stop translating that molecule of mRNA so that I synthesize the correct polypeptide. Don't want it to be too short, don't want it to be too long. If not, when it becomes part of an active protein, well, the protein's not going to be active. It'll either be inactive or it will be, how do you say, it'll function incorrectly. So, of all the codons in the codon table that you have here, okay, all but 3 code for amino acids, okay? The 3 that do not code for amino acids, these are simply called stop codons. Okay? I put it on your notes. There are 3 stop codons you are responsible for knowing. They are UAA, UAG, UGA. That's usually the order I memorize them in. Just UAA, then change that A to a G, UAG, flip the A and the G around, UGA. That's just the way it stuck with me. You will not find all 3 in the same molecule of mRNA. You will find one of these 3 stop codons at the 3' end of the mRNA. And when the ribosome, okay, reaches any one of these 3 stop codons in a single molecule of mRNA, it simply lets the ribosome know, stop translation. So the start codon is located towards the prime end. The stop codon is located at the 3' end. Simply put, start codon tells the ribosome where to begin translation, and the stop codon, any one of these 3, tell the ribosomes where to stop translation so that the correct polypeptide is synthesized. Now, technically speaking, when I say the ribosome reads a codon in mRNA so it knows which amino acid to grab, well, technically speaking, it's not grabbing the amino acids on its own. Okay. There's another type of mRNA called tRNA. The T stands for transfer. Okay. Why do we call this transfer RNA? Because, as it shows you here, tRNAs are responsible for transferring any one of the 20 amino acids to the ribosome. So technically speaking, okay, when the ribosome grabs an amino acid, it's actually grabbing it off of the tRNA. But it's up to the tRNA to help the ribosome translate the codons in the mRNA. And the way tRNA is able to translate, or I'm sorry, the way tRNA is able to help the ribosome translate the codons in the mRNA is through these. On one end of a tRNA, you will have one of the 20 amino acids attached. On the opposite end of the tRNA, you will find a base triplet. This base triplet is called an anticodon. Okay, so anticodons are base triplets that are found on tRNA. Listen carefully. For every codon that you find, for every codon that codes for an amino acid that you find in mRNA, there will be a tRNA with an anticodon that complements that codon. So all these codons here that they show you are coding for the different amino acids abbreviated with 3 letters. Keep in mind, everybody, going back to chapter 5, you had to know that 3-letter abbreviations. This is the reason why. You can see serine here. Okay. What were some of the others? Cysteine over here. Okay. Glycine over here. These were some of the ones you had to know. Okay. This is the reason why. So make sure when you review proteins, you also review the 7 you needed to know so you know what you're looking at here. So for every codon that could possibly be found in a single molecule of mRNA, you will have a tRNA with an anticodon that complements that codon. Okay. This ensures that when the correct tRNA that has an anticodon that complements any one of these codons in mRNA, this ensures that the tRNA transfers the correct amino acid. And I'll show you more of what I mean in a second, so it makes a little bit more sense. These are just different representations for tRNA. Now, they're not called tRNA because they fold up in kind of a T. They're called tRNA because they transfer amino acids. And what ensures that the tRNA transfers the correct amino acid is in the anticodon. Okay, and I'll show you, as I said, that in a little bit. Ribosomal RNA. Okay, I forgot to put it down. The R stands for ribosomal. This gives you way more than I'm going to give you. This is just to let you know there are different types of ribosomal RNAs. This type of RNA, it is called ribosomal RNA because it contributes to the formation of your ribosomes. Doesn't matter if the ribosome is prokaryotic or eukaryotic. All ribosomes are not only made up of proteins, but they're also made up of ribosomal RNA. And there are different types of ribosomal RNAs. The only time that I ever lectured on the different types was for advanced cell and molecular courses, okay, never for freshman or sophomore levels. But this is showing you much more. But okay, so what I want you to take away from this is that ribosomal RNA, okay, will combine with proteins to form your ribosomes. Okay, now there's something in between here, and I'll talk about that in a second. So it's very brief for ribosomal RNA. Okay, the last component I want to talk about in addition to the 3 types of RNA before we go over translation is your ribosomes. What does a ribosome really do? Well, first of all, structurally, ribosomes are made up of 2 ribosomal subunits. Okay, one is called a large subunit and one is called a small, and I'll show you on the figure. Both ribosomal subunits are formed by the ribosomal RNA and proteins. So both ribosomal subunits are formed by these 2 compounds here. Now, the ribosome's job, okay, is to play a role in matching the mRNA codon to the complementary, and I left it out, anticodon in tRNA. So I need to add an anticodon of tRNA, which they're showing you here. Okay? And I'll show you this a little more in a little bit. Okay, then what the ribosome will do is with the help of the tRNA, the correct tRNA based on its anticodon binding to the mRNA codon, that ensures that the correct tRNA transports or transfers the correct amino acid. Then what the ribosome will do is take that amino acid off of the tRNA and covalently bond it to another amino acid. Well, keep in mind, okay, when 2 amino acids are being covalently bonded, we call that covalent bond a peptide bond, and I'll show you that in a second. So here this is showing you again a little more detail. Ribosomes are made up of ribosomal RNA and proteins. These ribosomal RNA and proteins make up a large ribosomal subunit and a small ribosomal subunit. Okay. Once you become familiar with these subunits, then they just say large and small subunits. Okay. But you're going to hear me say ribosomal. Now, the only time the large ribosomal subunit and the small ribosomal subunit come together is during translation. Whenever translation is not occurring, these 2 subunits actually stay separate from each other. This is one way a cell is able to control when they do their job. Now, the reason for showing you this figure is to show you that when translation is going to occur, the large subunit will attach to the small subunit, and this will create 3 sites. You only see these three sites, or we only talk about these three sites when the large ribosomal subunit attaches or complexes with the small ribosomal subunit. These three sites are known as the E site, the P site, and the A site. Okay. Now the way these sites actually work is in the opposite direction. Okay, but when you're first learning these sites, for me, I would always say EPA because of the government agency, but the direction they actually work is the opposite direction. But for now, going left to right, the E in E site simply stands for exit. Okay, this is where a tRNA will exit the ribosome. That's the E site. Sometimes they'll show it to you in a figure, a lot of times they won't. I try to use figures that show it to you. The middle site is the P site. Now, technically, the P site stands for peptidyl. Okay, I like to think of the P site as standing for the P in polypeptide. That's what I like to think of the P site standing for. P in polypeptide, because this is where your growing polypeptide is located as the ribosome is synthesizing it. It's located in the P site attached to a tRNA, as we'll go over more. Okay. The last site is the A site. Now, the A actually stands for amino acid, but I like to think of the A as standing for access, ACCESS. Okay, the reason for that is because the A site is the site that allows a tRNA to access the ribosome, to enter the ribosome. But we already used E for exit, so we don't want to use E over here again. Okay, so the way these three sites work is actually in the opposite direction. And what is moving through these three sites? It's tRNA. So it's these tRNAs that are moving through these sites going in this direction. A tRNA that's transferring an amino acid to the ribosome will enter the A site. Okay. Eventually that tRNA will move into the P site where it will have the growing polypeptide attached to it while it's in the P site. Then eventually what happens is that growing polypeptide is removed from that tRNA and it'll move into the E site so it can exit. So notice here, when a tRNA enters the A site, access site, it's transferring amino acid, but by the time it reaches the E site to exit the ribosome, notice the amino acid is gone. The ribosome has grabbed it and used it to synthesize a growing polypeptide. So this is what we'll talk about when we get to the three steps. So the question is, well, what the heck is the ribosome doing then? I mean, the tRNA seems to be doing all the work. Well, no. What it is when that ribosome grabs the amino acid, okay, the amino acids that the transfer RNAs have transferred to the ribosome, what actually happens, okay, is that the ribosome will take the amino acid transferred by a tRNA and covalently bond one amino acid, okay, covalently bond one amino acid to another amino acid depicted by the plus sign here, okay. Once this occurs, okay, or when this occurs, what is actually being created is a covalent bond that we call a peptide bond. Now keep in mind, we're talking about organic compounds, so there are many covalent bonds, as you can see, throughout these two amino acids that have now been chemically bonded to each other. Only the bond, okay, attaching one amino acid to another, that's the only covalent bond that's given a special name, a peptide bond. You have to get used to this in the sciences, but mainly it has to do with what direction you're going in. But in the sciences, okay, we talk about covalent bonds being single and double, polar and nonpolar, but there's a lot more to covalent bonds, such as some covalent bonds are given special names, okay. In your Lab 8 lab, we talked about beta-mercaptoethanol breaking types of covalent bonds called disulfide bonds, which I'll talk about at the end of this chapter. Okay, then there are other types of covalent bonds that have to do with attaching one amino acid to another. These covalent bonds are called peptide bonds. So what the ribosome is doing is it's forming these covalent bonds depicted by the black lines up on here that you can see between the amino acids. These black lines are these covalent bonds here called peptide bonds. So that's what the ribosome is doing. So therefore, technically speaking, the ribosome is synthesizing the polypeptide. Okay. Now, it's possible, okay, for more than one ribosome to translate the same one molecule of mRNA. Whenever that occurs, we call it a polysome. So in other words, it's not necessarily a one-to-one ratio. One mRNA doesn't lead to the synthesis of one polypeptide. More often than not, one mRNA is translated by multiple ribosomes, leading to multiple polypeptides. Because when it comes to proteins doing work, it's not one protein doing the job. It's often a group of the same proteins doing the same type of specific work. And this is an actual polysome right here. Okay, so just like for transcription, translation consists of three basic, if you want to call them, substeps. And they go by the same name: initiation, elongation, and termination. Okay, so the first basic step of translation is initiation. This is the way it works for initiation. Initiation begins with the 5' cap of mRNA being used to attach mRNA to the small ribosomal subunit. Second, okay, the first tRNA, called initiator tRNA, will use its anticodon to base pair with the mRNA start codon. If you look up here in the circle, it's showing you. Here is the start codon, AUG. It's buried within a sequence called the Cossack sequence towards the 5' end of mRNA. They're not showing it real good here, but this is towards the 5' end. Initiator tRNA, it's the tRNA that's going to have an anticodon that complements the mRNA start codon. Therefore, this initiator tRNA, its anticodon is made up of the base triplets UAC. U base pairs with A base pairs with U, C base pairs with G. Okay, so the anticodon is the complement of the codon. The codon is part of the mRNA, the anticodon is part of tRNA, and this ensures that the correct tRNA transfers the correct amino acid. Third, the large ribosomal subunit will attach to this mRNA small subunit complex. When the large subunit attaches to the small subunit that has the mRNA attached to it, it will attach to where the initiator tRNA carrying MET, transferring MET, is located in the P site. This leaves available the A site for a second tRNA to enter. Okay. Now listen carefully. Once the large subunit has attached to the small subunit, which is attached to the mRNA and the initiator tRNA, we now have the formation of the translation initiation complex. This is the translation initiation complex. It's made up of both the large and small ribosomal subunits with mRNA, the start codon attached to the initiator tRNA located in the P site. This is a translation initiation complex. Once this complex has been formed, now the ribosome is ready to begin the process of synthesizing the polypeptide. Synthesis of the polypeptide by the ribosome, this is the second step called elongation. Okay, so the second substep or second basic step of translation is called elongation, and this is the synthesis of the polypeptide. The way it works, and unfortunately the figure I have here, it's sped forward a little bit. You can see here they actually aren't going straight into the next step. They've actually skipped a few steps. So notice we have a tRNA. Its anticodon complements the codon in the mRNA, so they're attached. But this tRNA is located in the P site. If you look at the opposite end, opposite for where the anticodon is, you can see we already have a polypeptide chain made up of 4 amino acids, each chemically bonded through a peptide bond attached to this tRNA. The A site is available for the next tRNA to enter. So when elongation occurs, the next tRNA will access the A site, transferring the correct amino acid. How does we ensure that the correct amino acid is transferred? Only the tRNA with an anticodon that complements the next codon of the mRNA will be able to base pair and chemically anchor itself to the mRNA. And now the ribosome knows, okay, this is the correct tRNA transferring the correct amino acid. From there, what actually happens is the growing polypeptide that is located in the P site, the ribosome will break the chemical bond holding this polypeptide to this tRNA in the P site. And it will take this growing polypeptide and now create a peptide bond with the newly transferred amino acid here in the A site, which they show you down here. Now, this tRNA that's in the P site that initially had this growing polypeptide, now if you look down here, it doesn't have anything attached. Okay? What happens next during elongation is that the ribosome will shift. The ribosome will shift, okay, towards the 3' end of the mRNA. I'm not even going to try to write that out. Of the mRNA. Okay, so again, the ribosome is the one that is doing the moving. The ribosome will shift towards the 3' end, or another way to put it is the ribosome will shift downstream to the next codon. So that is what they are showing you from here to here. The ribosome will shift down to the next codon that is now in the A site, and when the ribosome shifts down to the next codon, this tRNA in the P site that no longer has the growing polypeptide attached to it, that causes this tRNA to be moved to the E site so it can exit. It's done its job. It's through. Now this tRNA that was in the A site that now has the growing polypeptide attached to it, when the ribosome shifts to the next codon or towards the 3' end, that moves this tRNA in the A site over to the P site. Now the A site is available for the next tRNA to enter. Okay, so the ribosome will continue to shift down from one codon to the next, causing the tRNAs to move from the A site to the P site to the E site. Now what's happening to these tRNAs as this cycle continues, leading to a growing polypeptide, a longer and longer polypeptide being synthesized, is that the tRNAs bring in an amino acid, or they transfer a new amino acid. The growing polypeptide is attached to them. Then eventually what will happen is that growing polypeptide will then be attached to the next tRNA that transfers the next amino acid. And this guy that was here eventually ends up becoming this one over here that has no amino acids attached to it, and it'll exit. And this process will keep occurring, okay, with one tRNA after the other transferring the correct amino acid. And what ensures the correct amino acid is transferred? Only the tRNA with the anticodon that complements the correct mRNA codon in the A site. That ensures that the correct tRNA transfers the correct amino acid. This will continue to occur as the ribosome shifts from one codon to another, and elongation will not stop or terminate until eventually the ribosome will shift downstream towards the 3' end. And when it shifts down to the next codon, if any one of these three stop codons enters the A site, this is what will help trigger the third basic step of translation, termination. So I said before, make sure you know the three stop codons. These are the three stop codons. You will not find all three of these at the same time in the same molecule of mRNA. You will find only one of these three in the mRNA. And that's what they're trying to show you right here. We have a stop codon that has entered the A site. Listen to me carefully. In a eukaryotic cell, there are no, and even in a prokaryotic cell, there are no tRNAs that have an anticodon that complements an mRNA stop codon. So again, in both prokaryotic and eukaryotic cells, there are no transfer RNAs that have an anticodon complements any one of the 3 mRNA stop codons. So this ensures that no more amino acids are being transferred to the ribosome. However, there is a protein called release factor. Often release factor goes by the abbreviation RF. If you ever see RF but you see a small e before the RF, that just means it's eukaryotic release factor. They actually separate them up, but you can just know it as release factor for the test, okay? There is a protein called release factor depicted by this little yellow pill-shaped structure. This protein called release factor is the only molecule or compound that recognizes an mRNA stop codon. So when any one of these 3 stop codons makes up, okay, is located towards the 3' end of an mRNA and enters the A site of the ribosome, release factor is the only molecule that recognizes and binds to the stop codon. When release factor enters the A site of the ribosome and binds to that mRNA stop codon, it first triggers the release of the polypeptide from the ribosome, thus the name release. So again, when this molecule, a protein called release factor, enters the A site of the ribosome and binds to the stop codon, it first triggers the release of the polypeptide from the ribosome. Here they say free. Second, the release factor triggers the disassembly of this initiation complex. Okay, so second, release factor triggers the disassembly of the initiation complex. Notice the 2 subunits separate from each other. The mRNA is released. Thus, translation is terminated. Now, if you notice on some of these figures, they show you GTP and all that. Don't worry about that. You take an advanced course, they'll go over that. I'll have students come through this class and they'll tell me you're being too technical, you're giving too much information. But I've had several of those students come back after they take their upper level courses. They've come back and told me, no, it wasn't. We found out this is basic information. Okay? So just hang in there, study this. I've tried to give you the notes. I've given you videos. I hope you're watching the videos. You need to watch these videos for these last 4 chapters, please, okay? Any doubts about terminology that's used in a video or in the book, that's why I post the PDF and Word document notes for you. That's your safety net, okay? Go by what's there because I'm giving it to you in print. So when the ribosome finishes translation, what it has translated is the primary structure. In other words, it has synthesized the polypeptide, which is described as being formed or consisting of a long chain of amino acids that are all covalently bonded to each other or that are all peptide bonded to each other. So this is what the ribosome is doing. It's synthesizing the primary structure. It is synthesizing a polypeptide. Now the catch is this. If that ribosome that is performing translation is attached to the endoplasmic reticulum, thus if that ribosome is part of the RER, then that polypeptide that has been synthesized not only needs to fold up at least into a tertiary structure, if not a quaternary structure, depending on what type of active protein it's going to become. But if that protein is being synthesized at the rough ER, that protein or that polypeptide is going to become part of an active protein that is going to face tougher conditions, harsher conditions. Therefore, for only those polypeptides synthesized at the rough ER, they will undergo at least 2 post-translational modifications. One of these modifications is called glycosylation. The other modification is referred to as the formation of disulfide bonds. I put disulfide bonds, I'll edit this. It's the formation of disulfide bonds. Okay. First of all, the purpose of glycosylation. One of the purposes of glycosylation, okay, is to protect the active protein that the polypeptide becomes part of from being destroyed by your immune system. So this process called glycosylation, what does it involve? It involves attaching carbohydrates to the protein. Okay, now that protein may be formed by one polypeptide folded into a tertiary structure as they're showing you here, or it could be that active protein could consist of a quaternary structure made up of more than one polypeptide folded in a tertiary. But in either case, okay, glycosylation has to do with the attachment of carbohydrates to the protein, or to the protein that's going to become an active protein. In this case, it's showing you here. Now, in lay terms, the way we define glycosylation is, instead of saying carbohydrates, we simply say glycosylation is the attachment of sugars, okay, to a protein. This occurs in the rough ER, and then any protein synthesized in the rough ER, remember, it will be transported to the Golgi apparatus, where that protein will be further glycosylated. So glycosylation occurs in the rough ER, and it occurs in the Golgi apparatus, and then from the Golgi apparatus, that is where the Golgi will sort and separate the proteins and either transport them and package them into a lysosome, insert them into the cell membrane, or secrete them through a process called exocytosis. But in either case, one of the reasons or purposes of glycosylation is to protect the protein so it has time to do its work. One of the parts of your body it needs to protect it from is your own immune system. So these carbohydrates or sugars are said to function as ID cards. Second post-translational modification, I should have put it down, I should have added in formation of. Second post-translational modification is the formation of disulfide bonds. These disulfide bonds are often formed while the protein, or while the polypeptide that is transforming into an active protein, is still in the rough ER. And what happens, okay, is first of all, a disulfide bond, I hope I can spell this out, I forgot to put it down. If you recall, a disulfide bond is a covalent bond, but it's a special type of covalent bond. So another way to say it is, all disulfide bonds are covalent bonds, but not all covalent bonds are disulfide bonds. We don't need this here. So if you recall, in the lab, it talked about using a denaturing reagent called beta-mercaptoethanol, and it didn't say it broke covalent bonds. It said it broke disulfide bonds. Okay, these are special types of covalent bonds. So to say beta-mercaptoethanol breaks covalent bonds is incorrect. It breaks special types of covalent bonds called disulfide bonds. Well, what makes these special? Well, first of all, the purpose of forming disulfide bonds in the rough ER has to do with protein folding. If you recall, a protein shape is directly related to its what? Function. So this is the purpose of forming disulfide bonds. Okay, it's to help the protein fold into its correct shape. And these are very strong bonds because they're a type of covalent bond. Now, these disulfide bonds are formed between the R groups of the amino acid cysteine. Okay. So again, oh God. So what I'm trying to write down is formed between cysteine, or the amino acid cysteines. Put apostrophe S because it involves two cysteine amino acids. So basically what they're showing here is in a single polypeptide or between two polypeptides, wherever you have the amino acid cysteine, okay, if you remember the amino acid cysteine in its R group, for the amino acid cysteine in its R group, it has a functional group known as a sulfhydryl group. Okay, so when you have two amino acids along a single polypeptide chain or between two polypeptide chains, what happens is a cell will fold the polypeptide It'll either fold the polypeptide to where these two amino acids, cysteine amino acids, are across from each other. Therefore, there are groups. These sulfhydryl groups are across from each other. And this will allow other proteins in the cell to remove these hydrogens in these sulfhydryl groups and form a covalent bond between these two atoms of sulfur that are part of the R group of these amino acid cysteines. Therefore, it's a covalent bond, but since it's specifically between two atoms of sulfur that are part of the R groups of the amino acid cysteine, we call it a disulfide bond. The di meaning two, the sulf meaning two atoms of sulfur, but these are actually part of the amino acids cysteine. They are part of functional groups called sulfhydryl groups. Or what will happen in the RER is two separate polypeptides will be positioned to where the amino acids cysteine are across from each other. Therefore, their R groups are across from each other. And then therefore a covalent bond called a disulfide bond can be formed. The purpose of this again, protein folding. Well, if you look down here, okay, well, what's the importance in this? Insulin. Okay. Insulin is actually a protein. We call it a peptide hormone, but it's a protein, everybody. Okay. Initially, it's synthesized as a single polypeptide. And then what will happen is disulfide bonds will be formed to help it fold into the correct shape. And then something I'm not going to test you on is parts of it. Parts of what were once a single polypeptide are cleaved. They're cut to make it look like it's made up of two small polypeptides. But notice you have multiple disulfide bonds to help this molecule or help this polypeptide fold into its correct shape. In other words, help the protein fold into its correct shape to where now we not only have insulin, but it's active, it's functional, it's ready to do its job. Okay? Okay, now the last part here, when it comes to our genetic information, okay, in the form of genes, the DNA sequences that form the genes, they're very sensitive, all right? Therefore, if a mutation occurs, it could cause a big change in the protein to where either sometimes the protein is not even produced, or it's produced but it's not functional. It's like having a car without a battery. Or it's produced and it goes, in a manner of speaking, rogue. It starts causing problems. Okay? So the DNA sequence that forms a gene, it's very sensitive, to mutations. Now, not all mutations that happen to a gene lead to negative side effects on your phenotype. I mean, it's what leads to different alleles that code for different traits, such as dominant phenotypes versus recessive. It's just sometimes it can lead to problems. Case in point, sickle cell disease. Okay, sickle cell disease, first of all, it's a genetic disorder. Okay, it's an inherited genetic disorder. It's caused by a single base mutation. Okay, now the gene associated with this genetic disorder called sickle cell disease, the gene is known as the beta globin gene, and it's located on chromosome 11. It's a very large gene. Okay, it's made up somewhere roughly between 5,225,000 base pairs to 5,227,000 base pairs. Okay, very large gene. However, the genetic information, okay, that codes for the polypeptide, there are certain stretches of it that if it undergoes a mutation, it can lead to adverse problems with the polypeptide that is synthesized, and this is what I'm going to go into. What they discovered is that along this gene, the beta globin gene, okay, there is a stretch where you have a codon, okay, that should read in the forward strand GAA. Therefore, in the reverse strand, the complement is CTT, so that when this reverse strand is used as a template to synthesize mRNA, we end up with a codon that reads just like in the forward DNA strand, GAA. What's the importance of that? Well, GAA is a codon that codes for glutamate. Everybody, glutamate has an R group that is polar. It is hydrophilic. It is highly reactive. It likes to socialize and mingle. Okay? However, for those that have inherited the genetic disorder called sickle cell disease, what happens is both beta globin genes, located on chromosome 11, located on the maternal chromosome and the paternal chromosome 11, have a mutation that occurs in this stretch I was talking about. So now what happens is instead of on the forward strand, the codon reading GAA, now the codon reads GTA. Therefore, on the reverse strand, the complement is no longer CTT, the complement is CAT. Okay, it's just one nucleotide base change. But what happens now is when RNA polymerase uses this reverse strand as a template to synthesize mRNA, instead of the mRNA having the codon GAA, now we end up with a codon that is carrying a one base mutation, a single base mutation. It reads GUA. Doesn't seem like a big deal, but the problem is this mutation happened to happen in the genetic information stored in DNA that happens to fall within one of the codons in the mRNA that is transcribed using the reverse strand. Okay? If you notice, there's a big difference between the codon GAA and GUA. GUA codes for a completely different amino acid that is hydrophobic. It is water hating. It does not like to interact and react and mingle with other molecules or ion. This one is known as valine. This amino acid behaves very differently from glutamate. Its behavior is completely opposite. The simplest way is it's hydrophobic. So what's the big deal with that? Well, this beta globin gene codes for the beta subunit, which is one of 4 subunits that forms your hemoglobin. If you recall, I said hemoglobin is made up of 2 beta subunits and 2 alpha subunits forming a quaternary structure. The problem is, okay, you can see here, when the polypeptide is being synthesized in the red blood cell, which is the only one that can express these genes, the beta and alpha globin genes, what happens is for those individuals that have inherited a mutation in both beta globin genes from their mom and their dad, now what happens is that when gene expression occurs and translation occurs, the second step, we end up with a polypeptide that has the amino acid valine instead of the amino acid glutamate. Remember, this is all starting from a single base mutation. That single base mutation leads to a mutation in the polypeptide. Now have one incorrect amino acid forming part of the polypeptide in comparison to the normal. Now what happens, okay, is that one incorrect amino acid resulting from that single base mutation results in the beta subunit folding incorrectly. It doesn't fold correctly as they're trying to show you in a simple diagram up here. Now what happens? Whereas when the beta subunit, okay, when the genetic information coding for the beta subunit is correct, the beta subunit folds into its correct shape, then it complexes together with other beta subunit, another beta subunit, and 2 alpha subunits, forming normal hemoglobin. Now everybody, mature red blood cells don't have a nucleus. They get rid of their nucleus. Therefore, they have a lifespan of about 120 days in your blood. Now, why do they get rid of their nucleus when they fully mature? Because they want to have as much room as they can for these hemoglobin molecules. So red blood cells are described as being packed full of these hemoglobin molecules so that they can transport enough of the gas O2. But these hemoglobin molecules, despite the red blood cell being packed full of these hemoglobin molecules, these molecules do not stick to each other. Okay? Whereas if somebody inherits a sickle cell gene, a mutated beta globin gene that has that single base mutation, leading to a single mutation resulting in one incorrect amino acid, which in turn causes the beta subunit to fold incorrectly. And not just fold incorrectly, notice what's exposed? that hydrophobic amino acid. Now what happens is as these misformed or misfolded, incorrectly folded beta subunits come together, forming your hemoglobin molecule, now when the red blood cell produces many of these, what in turn happens is that these beta subunits, let me do that again. What in turn happens is the beta subunits on one hemoglobin molecule begin to stick to the beta subunits on another hemoglobin molecule. Therefore, in a manner of speaking, we have clumping or agglutination. Now, in turn, what that causes to happen is that since a red blood cell is packed full of these hemoglobin molecules, when these hemoglobin molecules are not formed correctly, when they're sickle cell hemoglobin, they start to clump together or agglutinate inside of the cell. And what you see happen is it turns and twists the cell, causing the cell to go from a biconcave shape, or if you're looking at it from above, a round shape, twisting the membrane to where now the cell has a sickle cell shape. Now remember, all started with the single base mutation within the beta globin gene, if you inherit it from both parents. I'm simplifying that too. That in turn leads to the one incorrect amino acid, which in turn leads to this polypeptide folding incorrectly, which in turn leads to the formation of a hemoglobin molecule that sticks to others, which in turn changes the phenotype of your red blood cells, causes them to become sickle shape. That in turn leads to clumping and possible blood clots. Your spleen and your liver are responsible for getting rid of old red blood cells as well as malformed red blood cells. But what happens is the spleen, while it's trying to get rid of these sickle-shaped red blood cells, it gets overwhelmed. It can fail. You can damage it. Also, likewise, your body, your spleen and your liver recognize that these red blood cells now have a sickle shape, so they start breaking down the red blood cells. This can lead to a condition such as anemia. In other words, you're low in O2 because you don't have enough red blood cells being produced. In turn, if you're low in O2, keep in mind, we need O2 for cellular respiration. Can't produce enough ATP. You start experiencing physical weakness, possible heart failure. But notice, all this starts from a single base mutation, which ends up having a detrimental effect on a person's phenotype, the offspring's phenotype. The average lifespan used to be 25 years of age, at least up into the 80s. I think I've read where now it's a little longer, but they're still trying to figure it out. Listen carefully, everybody. If you pay attention to the literature out there, you might have heard of a system called CRISPR and how scientists want to use CRISPR for gene therapy. Okay, well, this is one of the diseases they want to target using that CRISPR system. Why do they want to target it? Because they know they just have to correct, okay, one base pair mutation. They know they just have to correct it and revert it back to the normal base pair here that it's showing you. So this is why sickle cell disease, along with another genetic disorder, are being targeted for gene therapy using this CRISPR system, because it's not a large, okay, mutation to, how do you say, reconvert the gene back to a normal state that has to be performed. This also right here is one base pair. So when you're looking at your lab, okay, today, lab 9, day 2, you're going to hear me say the length of a DNA fragment is described in base pairs. This is what we mean. It's double-stranded and the bases pair together. Are there any questions? Okay, I'm going to stop